Hello, everyone. It is my great pleasure to introduce to you a distinguished scholar and brilliant researcher, Dr. Jinyuan Chen. Dr. Chen is an assistant professor at Martinez Center for Biomedical Engineering at Massachusetts General Hospital and Harvard Medical School. She earned her PhD from Stanford University with a major in electrical engineering and a minor in statistics. Following her graduation, she pursued further training in neuroimaging as a postdoctoral fellow at Martinez Center. Her lab advances neuroimaging technique with a focus on functional PET-MRI and ultra-high field fMRI and computation approaches to investigate biophysical and molecular mechanism underlying intrinsic brain activity and neurobiological consequence of cognition, arousal, and disease. I'm personally very interested in her research. I'm very excited for this talk. Dr. Chen, thank you for being here. And without further ado, I will let you take it from here.
Okay, great. Yeah, thank you so much for having me today and also the nice introduction. So I will just jump right in. So today I'm going to share some work from our lab where we leverage a new functional PET method and a PET-MRI to gain some new insights into brain dynamics. And I also have to say a lot of work I'm going to present today is still in a preprint and abstract form. So I view that as a great opportunity for us to receive some feedback from this audience too. So first, I feel probably for this particular audience, I don't need to reiterate how important fMRI has been a tool for us to understand brain functions, both in the task evolved states and also in the resting state conditions. But for people like in my generation or younger, we probably are not quite familiar that before fMRI comes out, PET has been the primary technology for mapping brain functions. So the basic idea is like the neural and glial activity or consumes energy when they are activated by a task. So in this case, like we can have two separate scans. In one PET scan, we kind of measure the glucose while the participants just doing some control baseline conditions. And then in another PET scan, the participant doing the task of interest. And then by mapping the metabolic differences between these two conditions, we can identify, which region have elevated glucose metabolism that are most relevant for this task. So this is the rationale for functional imaging using PET. But you can clearly see there are a lot of challenges. First, it's like the sensitivity and resolution is pretty low. And also because you involve two separate PET scans, so then there is a substantial inter-scan variability comes in. And then, but like this was like in the late 1980s, over years, there are so much improvement in PET hardware, like improving the sensitivity and the resolution. But on the other hand, there's also some recent kind of refinement of the functional PET methodology that allows us to overcome the limitations of two separate scans. So this was actually a technique first introduced by Jacob Hooker's group at our center. It's almost 10 years. So basically the idea is that for conventional PET, you have a bolus injection of the tracer, and then you have two PET scans. One is a task scan, one is a baseline scan, and you map the difference of the metabolism to identify the regions activated by the task. But now this functional PET FDG or FPET FDG kind of replace this bolus injection with a constant infusion paradigm. So in this case, because the tracer was slowly infused to the body at a constant rate, so the PET time activity curve will gradually reach a steady state. So where the slope of the curve is proportional to the steady state cerebral metabolic rate of glucose. So now we have a task. Then the time activity curve will gradually reach a new steady state, which will manifest as an altered slope of this time activity curve. So by tracking these changes in the slope, we can infer the dynamics occurring in the glucose metabolism. And then we can also map the metabolism in a way similar to fMRI under general linear model framework. just some comparison with FMI. So for FMI, we know their baseline is almost like flat, and then there is a stimulus-driven activity, elevated activity, and returns to baseline. But for FPET, the baseline is roughly linear line going up due to like the accumulation of FDG in the body, and then for the task-evoked activity or how it deviates from baseline, it's like increased slope, kind of indicating changes in its metabolic levels. And also it just got trapped, it wouldn't go back to baseline, it just like returned to a flat level. So that's the difference. And also we also need to notice the time scale differences. So for FMI, it's over seconds. For PET, while nowadays you can reconstruct over second level or even faster rate, but the metabolic process is tracking most robustly still over several minutes. So just to give an example, nowadays people have been applying to several different tasks, but I'm using the initial visual task as an example. So here the participants doing the experiments over 90-minute scan, and there are blocks of time where they're doing a flickering checkerboard. So if you look at control region in the frontal area, you can roughly see a linear line going up, the blue line here. But then if you focus on those visual cortex, you can see when there's a task, there's increased slope in the time activity curves. So this difference is most obvious if we take the difference or removing the baseline, or maybe for the remaining of my talk, I may refer to as detrending, and then you can more clearly see when there's a task, there's elevated change in the slope. So for simplicity, for the remainder of the talk, I will primarily showing these kind of slope changes or the detrended signal. When I first learned FPET, I found it so helpful to play with some sensory data, both shared by Jacob Hooker's group at our center and also some public data from Monash University. So I'm also show some of these raw time courses just for you to get a sense about the sensitivity and the variability of these FPET data. So for instance, these are some like a motor task over 90 minutes. And then these are from 7 subjects, like their mean and standard deviations of the detrended time activity curves in the motor cortex. What do you notice? Like it's quite noisy, but then on the other hand, you can track some like patterns following the task. And then not surprisingly, if you look at the activation across the brain, you can clearly identify elevated glucose in the motor cortex. And here are some data I play with from the public visual FPET data. Similarly, you will see how much variability it is across subjects, and then but you can still definitely track these task-induced changes in metabolism. This is at the group level. There are 2 things I want to point out. First is like, in addition to activation in occipital cortex, I also see elevated activity in the frontal lobe. So in general, I found this technique are also sensitive to some general baseline changes in brain metabolism, may related to attention of task engagement. So that's one thing. A second thing is that if you compare it to fMRI, you can clearly see that it's much higher sensitivity in the fMRI signals, even just one block of data. They got much higher statistical scores and compared to this FPET. So I feel it's helpful for me to get a sense of like how strong the signal is. But in any case, it really kind of gave me confidence about how robust this technique in tracking task-related changes in metabolism. So it's really an interesting technique. So why that could be helpful? First is like there are some cases we know like fMRI can be sensitive to blood flow changes driven by autonomic physiology. And there's some evidence that these pattern metabolism are less sensitive to that. So in those cases, FPET could be an alternative technique for functional mapping. And also it allows us to extend imaging to the body because our body organs consume glucose. And then compared to fMRI, it also have a nice trait of being quantitative. I think it's particularly valuable for clinical applications. But regardless of these advantages, like in terms of sensitivity and resolution, no way can replace fMRI. So I think a lot of people are most interested that it's just unlike fMRI, maybe more sensitive to flow oxygen metabolism, it does provide complementary information about glucose metabolism. On one hand, a lot of metabolic dysfunctions may occur prior to functional anatomical atypical patterns. So that's why it's interesting to look at glucose metabolism. And from neuroscience perspectives, as I just mentioned, because fMRI and PET-FDG really look at different facets of brain activity. And there are many cases, the flow and oxygen metabolism and glucose metabolism can decouple. And this has already been shown by several recent studies kind of investigating both fMRI and fPET. So for instance, this is a nice study looking at the resting state connectivity patterns. They reviewed distinct patterns of functional connectivity and metabolic connectivity. And here is another example where a study shows that you can identify different spatial patterns of glucose activation and bold activations, especially you don't see those deactivation in the fMRI in the metabolism. So fPET really brings a lot of exciting opportunities, right? So on one hand, we now can probe the relationship of glucose metabolism and hemodynamic signals under the past evolved conditions. And also in special cases, if we have very high sensitivity, we may be able to push the resolution in finer temporal resolutions. So we can link moment-to-moment changes in metabolic dynamics to ongoing brain functional dynamics. And on the other hand, this is really an emerging field. There are a lot of questions on how we can refine this experimental design, acquisition, and the statistical analysis of fPET, FDG. So these are those directions like our lab is really quite interesting nowadays. So for today's talk, I'm going to probably share some lines of work along each of these directions. And then due to some data dependencies, I will actually reverse the order. I will first share research where we leverage simultaneous EEG PET-MRI to look at the dynamic interplays of these electrophysiological, hemodynamic, and metabolic dynamics across like wake and sleep. Okay, so I think it's not surprised, a lot of studies has established that during sleep, there's drastic changes in a lot of metabolic processes. And even in human literature, they also observe big changes in the physiological processes. So for instance, people observe that during sleep, there's emergence of large waves of hemodynamic signals, and then they are locked through electrophysiologic signals and also drives CSS flow during sleep. But the question is like, there are so many processes going on in sleep, and then how are these processes coordinated in time? Especially nowadays with fPET, we can also have this insight of dynamic evolutions of glucose metabolism. How are they temporally tied to other physiological processes in the brain? And also for PET and fMRI, we have very high have whole brain coverage. So this also allows us to probe what are the metabolic and hemodynamic states underlying the differences of sleep and wake. So to look at these questions, we just have all three modalities together and try to track these electrophysiological, hemodynamic, and metabolic signals across wake and sleep. So this data were collected at the Martino Center using a 3T Siemens scanner with a brain pad insert. And we also use concurrent EEG measures to delineate subjects' arousal states. And all the scan took place in the afternoon. And then these are like functional PET protocol. We also add a slight bolus injection at the beginning to boost the sensitivity. And then we reconstruct the data about like 30 seconds per temporal frame. And for fMRI, we just limited by the hardware. It's very standard with no acceleration, just like conventional resolution acquisitions. Okay, first to showcase, we can reliably detect the effects of arousal modulations using all modalities. So this is one example and showing the spectrogram of an occipital EEG electrode. So we can use the power of alpha wave activity to infer the subject's arousal state. As you can see, at the beginning, we can see like clear alpha waves, the subject awake, and later they fall asleep. And in the later phase of the scan, they have a brief wake period, and then they fall asleep again. So if you look at the fMRI time course, just consistent with the previous study, you can see people fall asleep, there's emergence of large hemodynamic waves. And when there's a brief wake period, they really kind of diminish again. And for FPET, here again, I'm showing the detrended time course, and you can see when people fall asleep, there's a decreased slope in the time activity curves. And then when there's a wake period, it's subtle, but you can see there's a deflection in the time activity curves, and then goes down again when people fall asleep. And this is another example where there's variations of different stages of sleep of these particular participants. And then you can see in the different phases there are changes in the time activity curves. So basically, this suggests like we can have the sensitivity to track arousal-induced changes in all different modalities. So now going back to the first question, to look at the temporal coupling of all these different physiological processes. We just mentioned like for fMRI, a clear feature is like when people become drowsy or gradual descent to sleep, there's emergence of large hemodynamic waves. So that's why we use a time window measure to track these amplitude of these oscillations over time. And then to match the time scale of these fMRI signals to PET over 30 seconds, so the time window measures of fMRI signals are also over 30 seconds. So then we have a time window measure of this global fMRI signal activity. You can view it as a way to quantify the power of these fMRI oscillations over time. And then to link to PET, if you remember that FDG PET measures the track of the FDG accumulation. So it's a cumulative measure. That's why to mimic that property, we do a temporal integration of these fMRI signals. And then we do a low order detrending to remove those major baseline fluctuation. So we get this fMRI modeled FPET dynamics. These are just my first attempt, but I'm very surprised to see how well it can track these FPET time activity curves, which is shown in the purple color here. Not just in general sleep-wake transitions, but look at where my arrow points, even some subtle variations in arousal, it already tracks pretty well. And then these are single subject data. And if you look at the group level, we can clearly identify significant correlations between these PET and fMRI signals with emergence of large waves of hemodynamic waves. And then you can see a reduction in glucose metabolism. They're tightly coupled over time. So how about other physiological processes? So a lot of studies has already established that over the timescales of seconds, these fMRI oscillations are time-locked to EEG slow wave features, and also the CSF flow, as recently kind of shown by in the next study. So the question is like now we show over minutes, these fMRI oscillations are temporally coupled to glucose metabolism. Does that suggest these amplitude variations of other physiological processes like EEG and CSF flow over minutes, they're also coupled to metabolism? Indeed, we observe that, and this is work done with the help of my student, Dakota Javan Fine, and then we first characterize the EEG arousal measures. Basically, the idea we take the ratio of alpha wave activity versus delta and theta activity. It's kind of established way in several existing literature to track gradual changes in arousal. Here is just a case showing example here, so you can see the variability of arousal levels over time. And look at the time scale, it's over several minutes. And then for CSF, we follow the previous science paper, like we take the inflow contrast of the fMRI signal from the fourth ventricle, and then we track the amplitude variations over time. And then we link them to PET signals, just as what we did for the fMRI signals. In both cases, we can still see the clear, significant coupling of all these modalities. So this does suggest like, also not too surprising that during the descent into sleep, all physiological processes are tightly kind of coupled to each other. So next, let's look at the spatial patterns. So again, for fMRI, the features we look at is like the emergence of large waves of hemodynamic signals. So that's why we quantified during sleep the fractional changes in the amplitude of these fMRI oscillations compared to wakeful conditions. And this maps the fractional changes of these oscillations brain-wide. You may notice that in the sensory region, like they really show the largest like slow oscillations during sleep. And then in metabolism, we quantify the ratio of the FPET time activity curves during sleep compared to wakeful condition. If you remember, if they reach steady state in each condition, the slope is roughly proportional to the cerebral metabolic rate of glucose. So using this as a way, we can see which region showing the most metabolic reductions during sleep. So we noticed that for sensory region where they're showing the largest oscillations in hemodynamic waves, they also have the lowest reduction of glucose metabolism. And by contrast, those default mode network area where they don't seem to have such a big increase in hemodynamic waves, they have the strongest reduction in glucose metabolism. So what does this imply? So this basically suggests there are network-specific hemodynamic and metabolic patterns during non-REM sleep, right? The sensory region remain active while those higher order regions are suppressed. So this is just a cartoon illustration. So fMRI reflects the slow oscillatory activity, and then we can see sensory region during sleep, there's a large waves of these oscillations. In contrast to default mode network, the oscillations are small. And because PET measures a very slow scale baseline activity, so in these sensory regions, while they have large oscillations, they also have a kind of slightly higher baseline activity compared to the default mode network. So what might be implications of this kind of network-specific activity? The possibility we are thinking about is related to a pathway that facilitates the sensory arousal during non-REM sleep. The idea is that there are several recent evidence from rodent study. They're showing that, for instance, like this is a LC activity during sleep. They also noticed that for the LC activity, when the rodent goes from wakeful states to the sleep states, there's decline baseline activity. But they also have these large around 0.02 hertz burst-like oscillations or burst activity in this LC activity. And this has been related to high activities promoting transient arousal, while low activities prepare entries into deeper sleep. And then we feel like this may be related to our findings is that if we plot the spectrum of these fMRI signals during sleep, in our data, we really see it's also peaking at around 0.02 hertz. And then consistent with some rodent studies, we also see the fMRI oscillations are more strongly correlated with the sigma band activity. So that has been related to oscillating behavior of alternating between a high arousal state, more attending to external stimuli, and an offline state, more concentrated to memory and internal processing. So that's why we interpret our results providing the brain-wide patterns that kind of sensory region have a slightly higher activity, and then that can facilitate potential sensory arousal during sleep. Okay, so let's make a short summary of this first study. And then first temporally, we using simultaneous EEG PET-MRI, we really show over minute scale this global metabolic, EEG, hemodynamic, and CSF dynamics exhibit a tight temporal evolution during the descent into non-REM sleep. And I found this particularly interesting is that a lot of existing studies mainly investigate changes in these patterns in isolation. So this basically suggests that their temporal dependence may also be interesting biomarkers for us to understand disease mechanisms. And for especially, we show there's network-specific hertz and hemodynamic activity. Particularly, the sensory regions remain metabolic active and exhibit those large 0.02 hertz periodical oscillations during sleep, whereas those higher order cognitive regions are suppressed. So this could be a pathway that facilitates transient sensory arousal or being awakening or alerting during like the sleep periods. And finally, because I'm from a method background, I also find it quite interesting demonstrating there's a potential for us to leverage simultaneous EEG PET-MRI to really look at those dynamic neurohemometabolic states underlying a broad spectrum of cognitive and consciousness conditions. Okay, so this is a particular example because sleep is such a big effect. So that's allowing us to look at a coupling in very high temporal resolution. But in general, as I scale back, like PET just provide ways to look at functional changes in glucose metabolism, and then FMI allowing us to look at functional changes in hemodynamic activity. So there's still lots of interesting questions we can address by relating them. And the second study I'm going to share from our group is how we can leverage functional PET-MRI to probe the relationship between glucose metabolism and complex network metrics. So complex network measures of brain activity is quite an active field, and then they try to summarize the brain activity with a few biophysical meaningful measures. And this typically involves populating brain into different regions and then compute Pearson correlations based on the pairwise temporal synchrony of these fMRI activities. And then there are a lot of interesting metrics we can extract from these interactions of brain regions based on the binary connection patterns. So for instance, like network hubs, referring to those nodes have tight connections with a lot of other nodes in the brain. And also like efficiency measures kind of quantifies the shortest path of one node connects to the other. These kind of measures are derived from complex network theory, so they may not necessarily have direct relationship with physical definitions of energetic costs. But because they kind of have shared terminology in describing the brain activity, so naturally we might be interested in relating these complex network metrics to energetic costs. So for instance, do hub regions have the highest energy demand? And also, does high global efficiency correspond to low energy consumptions? These questions might be important for us to reach appropriate interpretations of brain functional architecture derived from these complex networks, especially when we're trying to guide medication, and it's kind of important for us to understand what these metrics really means. So indeed, there are a lot of study already been done along this direction. And then they mainly based on the conventional static PET method and also fMRI measures of brain activity. And we know that conventional PET only has one snapshot of cerebral glucose uptake. So how do we probe their correlations? So some studies have leveraged the large samples along space. So basically, for each brain region, there's a measure of glucose and a measure of degree measures, and then they look at their associations. And then degree just a very common one of these network behavior people investigate. And some other studies will leverage the different samples across participants. So they track how these glucose uptake and then these degree measures change across participants and to probe their associations. And interestingly, these studies like kind of point to a roughly positive relationship between glucose metabolism and degree centrality. So basically, those regions have a higher degree centrality tend to have a higher glucose metabolism. But this approach usually leverage samples across space or subjects. So their association may also be driven by some anatomical or intersubject variability in those sources. So an alternative way to think about their relationship or probe their relationship probably could also be introducing a task or anything to make the system deviating from resting state. And then we track under this altered condition how glucose metabolism and these degree measures change. Do they exhibit consistent polarity of changes to probe their associations? So a student actually in our lab is interested in this question. So because we have this functional PET-MRI data, so we wonder, oh, maybe like task is a very natural and straightforward way to make the brain activity deviating from baseline. And then we can use PET-FDG to track the dynamic changes in the glucose metabolism, where you can use both fMRI to track the changes in degree centrality from the task compared to resting state condition. And then we can check the signs or whether the task-induced changes in glucose metabolism align with task-induced changes in degree centrality. So we kind of trying to assemble all the PET-MRI data we could have, including the sensory data I showed you before. And she also just chose a subset of the sleep-wake data I just showed in the first study. And then we also collected some new data in the working memory condition, just involving some higher order cognitive tasks. While we only have 10 participants, I think our results already quite consistent with a very nice recent study like published a few years ago, where they show during working memory task, the glucose uptake changes and fMRI signal changes. So you can see there's a very nice correspondence of our results and theirs. That's why we kind of using a relatively small sample size for this study too. And then in addition to task, disease is another potential case where we can probe the relationship between glucose metabolism and degree centrality when this whole brain activity deviates from normal baselines. But in this case, we don't have functional PET data, so Shirley came up with this nice idea to perform a meta-analysis of existing studies. Basically, these studies are using a conventional PET-FDG method, have a general measure of these metabolic levels in the resting state compared from disease states to the healthy controls, and also have the degree measures. They are just separate studies. But we can record regions demonstrating the most significant alterations of glucose metabolism, degree centrality, and then we can integrate them together. So here I'm just showing the results. Let's first look at the coupling of glucose metabolism and degree centrality across different task or arousal conditions. Here, just to simplify this results, we kind of also include sleep as part of the task. So the first row here shows regions, the spatial patterns where there's altered glucose uptake during task compared to rest. And the second row showing regions like how their degree centrality change during task compared to the rest. And if we plot these two metrics against each other, we found that quite interestingly, if you look at specific task, you may see a strong association. So for instance, like in the finger tapping task, you can see a significant negative relationship between glucose centrality and the metabolism. And sleep, similar to here. But then in working memory, you instead see a strong positive correlation. So there's an inconsistency in how they manifest in different tasks. And similarly for disease condition, here the first row showing which regions showing the most significant metabolic changes compared to control condition, and the red showing positive probability and blue showing negative probability, basically reduction of glucose metabolism. And then here shows the degree centrality differences. If we plot them against each other, you can see like for AD, depression, Parkinson, there seems to be a slight positive relationship, but in schizophrenia, the relationship is slightly kind of non-significant or negative. So again, it's not quite have a consistent pattern showing across all these disease conditions. So if we integrate all these brain puzzles and conditions in a pie chart, this is most clear at the group level, right? So for instance, if a parcel has an increased glucose metabolism, we count a fraction of them having significant positive changes in degree centrality and negative changes in degree centrality. It's unclear there's strong differences between them. And it extends to other cases where there are no significant changes in glucose metabolism and also a reduction in glucose metabolism. So there seems to be quite complex relationship between these measures. And if we scale back, it's not too surprising. These relationships are just not clear. Yeah, there are several ways we think about it. The first way is like, because these measures by definition are complex, right? For instance, if a task like our working memory task, it engage increased metabolism across multiple networks. So some nodes will also tend to connect more across networks for more integrated information processing. So in these cases, increased glucose metabolism may correspond with increased degree centrality. But in those sensory tasks where we kind of activate more focal regions, so these activated nodes may actually lose some connection with other networks or even within the network kind of showing a reduced connection with other brain regions. Additionally, a lot of measures in some conditions, how we return these connection purely based on a set of thresholds. So some cases, even a lot of nodes have elevated glucose uptake, higher activity, their connection gets stronger, but the number of connections after thresholds remain unchanged. So in those cases, these measures of glucose metabolism, degree centrality can deviate from each other. And also, if you look deeper into the signal mechanisms, so the fMRI signal kind of really relates to flow and oxygen metabolism, and the FDG is kind of glucose metabolism. And for instance, for excitation inhibition and also related to aerobic glycolysis, this glucose metabolism and oxygen metabolism may not refer identical patterns. So we just use a degree centrality as an example, but there are so many other complex network metrics of the brain. So Shelley also look at several other measures. They are tightly correlated to each other by definition by several ways. But if you correlate these metrics to the glucose uptake, and then each column here just shows analyzed results for different task conditions, you can definitely see there's some variability about how this relationship might alter between the different tasks. So fundamentally, this just suggests the results may not be, or the association may not be that simple as the current results published just purely based on those resting state measures. So to sum up for this second study, while we can identify significant correlations between glucose metabolism and degree centrality if we focus on specific task or disease, that kind of reflect interesting questions about the task and disease modulation. But in general, it's hard to link the definitions derived from this complex metric and those physical measures of energetic costs. We come to this conclusion by synthesized data across a lot of tasks and also a meta-analysis of disease states. And then while we just focus on degree centrality, and this complexity also extends to other complex network measures. So the message we try to convey is just like really it's quite complicated to link these abstract definitions of complex measures to true brain energy costs. And there are a lot of reasons why it's hard to make this association. Probably we just need to be more cautious when we're interpreting this graph theory or complex network measures of brain activity. Okay, by far, I think I'm more like conveying FPAT as a robust technique to track brain activity across different tasks and arousal. But for the final tag, I also want to share some considerations for the FPAT analysis. I just hope to give you a flavor that this field is really at its infancy. There are so many open space for improvement in its methodology and data analysis and potentially experimental designs. So as I introduced at the beginning, nowadays people analyze FPET based on a general linear model similar to fMRI. And in fMRI, we also model the baseline of activity, usually use lower order polynomials. But then it wouldn't be a huge concern because in general, those task-induced activity are at a much higher frequency compared to the low baseline. So in cases if we didn't fit the baseline activity wall, it wouldn't have such a big impact on the inferred task activity from the general linear model. But for FPET data, the baseline is kind of roughly linear. And then the task regressor, the major contrast is still linear, while we can also analyze it. But in general, basically this suggests that if we don't fit the baseline wall, a lot of those residuals are going to interact with your task regressor, kind of impacting your statistical inference. So in existing FPAT literature, people use different ways to model the baseline. They can use lower order polynomial, or they focusing on global signal, or with a variation by fitting the global signal using lower order polynomials, or they restrict the global signal to prior knowledge of tasks in active regions. But the FPAT signal is actually more complicated because before they reach a steady state, there are quite dynamic phases for the tracer to reach equilibrium. And this can be more complicated when we inject a bolus at the beginning to increase the SNR at the beginning. So basically, it's hard for these models to capture the shape very well. Just for you to get some sense of the idea, we demonstrate the residual patterning post-baseline removal using resting state data. So again, this is some open data shared by Monash University. In this case, because there's no task, and then we can see what the residual pattern looks like after you applying all these different detrendant methods. So what I'm showing here are 3 different ways of people removing those baseline, either mean across whole brain signal, like second or third order polynomial detrending. And then the different curve here shows the results of one single subject. I believe here have about like 26 participants. And then what's interesting is like we also perform a simulation just purely based on some empirical values of the kinetic parameters of the tracer. And you can see the simulation very well predicts these kind of group mean patterns, suggesting that this could be used as a resting state measure and generally reflecting the tracer patterns. So the question here is like, what if our task correlate with these residual patterns? So for instance, here just some examples of 3 cases we pick with different stimulus timing. And then the black curve are the residuals using the third order polynomial as an illustration. And then here just shows detrended task evoked effects. So these are 3 illustrative cases where the residuals have modest, medium, and very strong correlation with task regressor. So in this case, it's going to impact our inferences, right? There are 2 things we need to be aware. One thing is like, even it's a tiny effect, if we have a very large sample size, it's going to be significant at the group level. And second, there are a huge anatomical variability across the brain. So these impact are not consistent across brain regions. So it's going to show spatial patterns across the brain. So to show this idea, we just apply those sham task regressors to the true resting state data, because in this case, there are no actual task. So we just presuming there's a task going on and see what the spatial pattern looks like. And then again, there's 26 participants using the resting state data shared by Monash University. And then here it shows for different 3 regressors. Strikingly, you can already see significant activation patterns and different for different regressors. And then even just for mean tag, while globally it seems like the global mean can remove these residual patterns, but because of anatomical variability across regions, you can see different spatial pattern pumping up after you apply these sham regressors to that. So this result does suggest it's important to consider these potential baseline misspecification in F-PAT analysis. Another thing I want to quickly just go over is about these non-consistent variance over time for PET measures. I'll never be able to pronounce the word correctly. I think heteroscedasticity. So basically, the idea is like, because PET measures are count-based, so over time, as you infuse more tracers, there are more counts over time, and then the noise exhibits a Poisson-like property. So that's why the variance actually changes over time. Unlike fMRI, it's roughly stationary, and people are using autoregressive models to model these variance. So here you can see like the variance kind of increase as the mean time activity curves goes up. There are very standard ways to correct for these non-consistent error amplitude over time. For instance, people can use the robust error measures. So basically, there is no change how you estimate this Beta coefficient. It's just in the statistical inference, you account for these robust errors. And the second way is using weighted least square. You weight each sample by its variance measures. So its property is kind of well known. In the case if you have perfect modeling and then the weighted least square accurately reflect its variance, it's going to have benefits of improved efficiency compared to the robust error approach. But in cases where you have like pool fittings, it has a potential concerns of exacerbating this bias. So the basic idea is like if you have good model fitting, you should vote for the weighted least square case. If you have a case that your data have low sensitivity, and then not confident about your modeling, and then you should go with the robust error approach to mitigate the bias. So this is just an example. In the sensory case, we have a pretty good handle about the model baseline and these tacit vote effects. So you can see compared to the ordinary least squares and then weighted least square fitting squares that can increase the statistical scores compared to the finger tapping the visual stimulus. So these are the questions like my student Sean is pretty interested in pursuing. So, oh yeah, I will just highlight those brain regions. So a summary of like the third part, here are just some illustration of some potential concerns we need to be aware by extending GOM analysis to totally new data sets. So first is like baseline fitting now become very important. And the misspecification of the baseline could introduce bias in the measure and activation patterns. And the bias pattern really depends on the interaction of lots of factors like task paradigms, the trending approaches, and injection paradigms and tracer kinetics. And then even very tiny effects, when you have a large sample size, it's going to manifest at the group level, as we already learned from the lessons of fMRI. And but there are a lot of ways we can mitigate the bias. I won't go into the details, where if you're interested, we have a preprint and now in the revision phase, just go over some thoughts we think could help mitigate this bias. And another consideration is like how to reach valid statistical inference of FPAD statistics. So if you have a good idea of the model fitting, usually in the case when your data have high sensitivity, low baseline bias, and long duration stimulus, we can consider you incorporating those weighted least square. And in cases where you know your model has low effect size and poor model fits, robust error may be recommended. So again, these are not kind of very kind of exciting new technology or things. It's just like some subtleties when we need to think about or be cautious when we start to handle new data sets. Okay, just to wrap up, I hope I first convince you why I'm so interested in FPET these days. It's really a robust method for mapping brain changes in glucose metabolism. And then it will bring a lot of exciting opportunity for us to allow to interrogate the potential associations of metabolism and hemodynamic signals, and with deeper insights potentially relating to things like opportunities, for instance, like looking at the inhibitory excitatory balance and also aerobic glycolysis that direction. But from the very basic level, we are showing examples like when you have very strong effect, high sensitivity, you can really look at the time resolved interactions of these hemodynamic, metabolic, and potentially electrophysiological signatures across wake and sleep. But in general cases, it still gives us a very fresh perspectives. A lot of questions if we investigate purely on baseline resting state. It may be interesting to see how well they generalize to altered conditional brain states. And then functional pattern MRI is a pretty powerful tool for that. And finally, I don't want to end with negative. I think it's an exciting opportunity for people interested in statistics and signal processing to go into this field because there are a lot of open questions. And I'm now just touching one aspect about signal processing, but how to optimally design these techniques, there are a lot of opportunities going there. Okay, yeah, that's all my talk today. And I really want to appreciate all my colleagues, mentors, and my students who kind of contribute to the work I'm presenting today. And also open to any questions you might have. Okay, thank you.